Does everyone hear me? Okay. We're going to get started again. And first, I wanted to acknowledge some of the people who've helped orchestrate this conference. Joanne Nabrakowski and Carol Gregory. They're in the back of the room. And I can attest to the fact that they spent many long hours, overtime, et cetera, putting together folders, compiling lists, and making this conference a success. So thank you both. Also, I wanted to acknowledge some new people in our clinical group. As I mentioned, we have a new nurse practitioner who is helping tremendously with the clinic, Carol Freeman. Carol, could you stand up? We also have a wonderful clinical trial coordinator, so in the future, if you're interested in trials or hear about something new we're doing and have some questions, Judy Rodhoff, she is out of town today, but I wanted to mention her name. And I think many of you know Kathy, our spectacular nurse, who is going on to study to become a nurse practitioner, and our hope is that she's going to come back and join us in a couple of years. Kathy? You know, one of our big missions is education. And we are very dedicated to training the next generation of MS clinicians as well as MS researchers. In a few moments, I'm going to talk to you a bit about the research program and introduce you to some of our young scientists. But I also want to acknowledge some of our patients who've helped with the educational program. So we have patient conferences in which medical students and residents learn about multiple sclerosis. And some individuals from our clinic have volunteered their time and come to be examined and to discuss their disease to help educate new doctors and scientists. And they've been very courageous in fighting against their disease and have made great progress, and they're able to share that optimism as well. And I wanted to acknowledge two individuals who are in the audience. First of all, Angela Lofton. Angela has a condition related to multiple sclerosis called neuromyelitis optica, or NMO, and she's been fighting against the disease, and I'm happy to say has made great strides, and she's been very generous in helping to share her experience with our young trainees. So we have her certificate here to thank her, and I'm just going to move off the stage to give it to her. Thank you, Angela. Another individual I'd like to acknowledge is Chad Balema, who has been fighting against multiple sclerosis for a number of years, has a very inspiring story as well. He has made tremendous strides through hard work and dedication. I'm going to ask him to come up, and he may want to share a few words. But Chad has helped us numerous times in terms of coming to the university, and he lives a distance away, to participate in our patient conferences and other fora by which we train the young doctors and researchers. So, Chad.
I saw it move. Okay, sorry. I just wanted to say thank you to Dr. Segal and the MS team and for all their hard work and research and all the researchers that are out there, and the Holtom-Garrett family for continuing their support. I can't thank everybody enough for all that they're doing, especially for the secondary progressive MS. We all know, like we heard the earlier speakers say, there's not a lot out there. And whatever I can do to help, that's what we're there for. And like David Garrett said when I was speaking with him earlier, don't ever give up. We're in it for the long haul. So we're always going to be there. So Dr. Segal, if you need us, we're always there anytime you need us to help. And if there's anything we can ever do, just keep us in mind, and we're going to keep pushing forward. And I wanted to mention, because of Dr. Segal and the MS team, some of the therapies that I've been on, I have, my family's sitting right there. I have 2 young girls, they're 9 and 11. And this past spring, I was able, for the first time in 12 years, I was able to go on a walk around our block, which is about a mile long, with using just my cane and not a walk or anything. So thank you, and have a great day, everybody.
Thank you so much, Chad.
Thank you.
So this is what it's all about and what makes all our efforts worthwhile. Chad has made tremendous strides. There's some, research innovations we're trying to make, and particularly in progressive multiple sclerosis. And Chad has participated in some of our studies, and we're hoping to expand them to other individuals over the next year. So now to go on to talk a little bit about our research program. So we have a very extensive research program at the University of Michigan Multiple Sclerosis Center that spans many different fronts. Our ultimate goal is to get better at diagnosing multiple sclerosis and treating patients, and ultimately reversing damage and leading to better clinical outcomes. That's what the research is all centered around. We do this in many different ways. We use animal models, which means we can induce an MS-like disease in mice, for example, in order to better understand how the immune system attacks the nervous system. And that gives us insights into developing new drugs and strategies to stop the disease. We also have studies where we're examining blood from patients with MS to better understand how the immune system goes awry, and also to develop tests that will help us to predict perhaps the clinical course, to predict what therapy may be most suitable for an individual with multiple sclerosis, because not everyone responds to the same treatment. And ultimately, this culminates in clinical trials of new drugs and the development of new agents. And we have designed a number of new drugs ourselves, and have some patents on them, and we're trying them out in mice, and it looks very promising. So this is just a list of some of the individuals involved in our research. We have graduate students, young people who are studying for their PhD in neuroscience or immunology, and they're all dedicated to MS research and finding that new treatment, making that next advance. We also have postdoctoral fellows. These are scientists, young scientists who've already been awarded their PhD and are doing further study in our laboratory to perfect their techniques and to increase their knowledge, also dedicated to multiple sclerosis. And many of these individuals are involved in very basic studies that will hopefully elucidate how the disease is started, how it evolves, and once again lead to new treatments and tests. A little bit closer to the clinical trials of the human immunology research that we've been engaged in. And we are once again trying to understand how we could use blood tests to better manage patients with multiple sclerosis. And we're engaged in a number of multi-center clinical trials that Dr. Braley mentioned, but our ultimate goal is to start our own trials with our own agents over the next several years. So the clinical trial study program is robust and growing. This is really a program that's maturing. It's only 6 years old, but there have been tremendous strides made. Tiffany, Dr. Braley has done a great deal to build up the infrastructure for our clinical trial efforts with Judy Radhoff, the clinical trial coordinator. Our new clinical fellows who you're going to meet, Dr. Robert Pace and Dr. Jessica Stolk, are also getting involved in helping to build that. And then a number of us are working on these studies to help develop better tests for blood tests as well as MRI tests, so we could more accurately measure the amount of disease someone has and detect the new inflammation. So we also collaborate with a number of radiologists, and we're working on very sophisticated new ways of imaging the brain and spinal cord, and we're also studying the immune system from the patients to try to use both modalities together so that we could, to the benefit of our patients. And it all starts with these animal model studies in which we simulate multiple sclerosis in mice and try to understand how the immune cells get activated to cause the damage, how do they get into the brain and spinal cord, how exactly do they inflict damage to the myelin sheath, which is destroyed in multiple sclerosis, and the underlying nerve fibers. And as we understand more, then we could come up with new strategies to prevent the damage and hopefully reverse it. So I'm going to ask some of our young researchers to share with you in their own words what they're doing. They've assembled in the second center row here, and I'm going to ask them to stand up, and I'm going to introduce them. If you could come to the stage. Oh. We have a new arrival here. All the way from Ireland he came. So this is Dr. Julie Rumble, who's one of our postdoctoral fellows. This is Dr. David Giles. He's a brand new MDP. Oh, not yet. I'm jumping the gun. This is David Giles. He's an MD PhD student, so he's going to be a physician scientist, and he's joined our lab to understand MS better. This is Josh Stolman. I almost said doctor as well. He is another graduate student, a PhD student, as is Patrick Dunker. Another one of our postdoctoral fellows is Kevin Carbajal, who joined us from California a few years ago. And then Dr. Stephen Lawler, who joined us from Ireland a number of years ago. And Dr. Amanda Huber, who came to us from Mount Sinai Medical School. So I'm going to show you a couple of slides illustrating their work, and I'm going to ask them to make a few comments. So we're going to start off with Dr. Stephen Lawler.
Hello. So my work is based on one particular cell, a type of white blood cell that we believe to be pathogenic in multiple sclerosis. It's called Th17 cells. But basically, I followed these cells in a mouse model of multiple sclerosis, and we're attempting to see how these cells are able to induce disease. So what we can do with these cells is we can label these cells and we can transfer them into naive mice, and then these mice develop signs of disease very similar to the signs you'd see with the multiple sclerosis patients, like weak hind limbs and some forms of paralysis, and like Kevin will get to, often some visual impairment and things like that. But when we track these cells, we can see, if you see in the figure here on the left-hand side, you can follow these cells. We can label them in many different ways. These are just green labeled cells. And when the mice begin to develop signs of disease, we can find these cells in the brain and the spinal cord. And that's where they mediate the damage to the myelin that surrounds the nerves. But this takes a while for the cells to get there, and the cells have to enter the brain and the spinal cord from the blood, and how they learn how to get into the brain and spinal cord is really what we're trying to determine so that we can stop this from happening. And so what I've done is I also track these cells as they circulate through the body before the onset of clinical signs of disease. And I actually find that these cells accumulate a lot in the lungs and in the gut before they get to the brain and the spinal cord. And this is very interesting because this is where most of the bacteria that live in our bodies are actually found. And there's a new hypothesis that these bacteria might provide a trigger to stimulate these cells in genetically susceptible people. And so in order to determine whether the bacteria in these tissues might have an effect on how these cells get to the central nervous system, to the brain and to the spinal cord, we transfer these white blood cells to mice that don't have any bacteria. We call them germ-free mice. And basically, when we transfer these cells into these germ-free mice, we find that very few of these mice actually develop clinical signs of disease, and the ones that do get a much milder form of disease. And this is shown here on the right-hand side in the purple bar. And so it seems to us that perhaps the bacteria in the gut and the lungs and perhaps elsewhere do play a role in the initiation of MS-like symptoms in our mouse model. And this will be interesting, obviously, because we could perhaps modulate this using dietary alterations or using antibiotics as future therapeutic options for the treatment of MS. So this is where my research is going.
Thank you, Stephen. So I think that's very exciting, understanding how these bacteria that are a natural part of our bodies residing in the mucosal surfaces, which include the gut and the lungs, could predispose someone to develop multiple sclerosis. And as Stephen said, we may be able to change someone's diet or treat them with antibiotics by mouth that alter the composition of the bacteria to create a beneficial therapy. And I'd also like to mention that Stephen was awarded a prestigious fellowship grant from the National Multiple Sclerosis Society for this work. And Julie?
Thanks, Ben. So the work that I've been doing in this system is I'm also working with the mice, which I have a cute little picture of a mouse to sort of illustrate exactly what we're talking about. And I have the numbers on here because we use a scoring system. And basically, as the mice get sicker, the score gets higher. And so they start with a weak tail, and then their hind limbs get weak, and so on and so forth. And so the cells that I am focusing on with my work are called neutrophils. They're one of the most abundant immune cells in the body. And they respond to infections. And so they haven't really been looked at in the studies of MS all that much. They've mostly been ignored. But we think that they actually play a role. So we found some mice that are severely neutropenic, which means that they do not have very many neutrophils. And you can see that in the bar, let's see, down here. These mice, the blue mice here are normal mice. And then the red mice are mice that do not have neutrophils, as you can see. And so we induced EAE in these mice. And as you can see, the blue bar, the control mice, got sick normally. However, the mice that do not have neutrophils were highly resistant to disease. So we were very excited by these results. And we're hoping that maybe being able to modulate the function or the migration of these neutrophils might actually be able to become a therapeutic modality.
Thank you, Julie. So as many of the other speakers mentioned, the damage of multiple sclerosis is caused by immune cells that get into the brain and spinal cord and then create havoc. Well, as Dr. Braley, Dr. Maldreyer mentioned, people have been focused on certain immune cells called the T and B cells, and many of the new therapies are centered around either depleting those cells or interfering with their function. But this is another completely different immune cell that we think actually causes a lot of the problems. And so Julie's work could lead to a completely new class of therapies that work differently and may even further magnify the effects of some of the other newer drugs that are already established if they're given together. So, Josh?
Hello, everyone. So my work centers around looking at a very complicated portion of MS, and that many people have different symptoms, and certain people develop different clinical issues, and we think that this is related to where lesions develop in the central nervous system, but that's something that really needs to be studied a little bit more, and as we get better at figuring out how cells traffic to different areas of the central nervous system, we might be able to give more targeted therapies and maybe see where therapies work better in certain subsets of MS. So what I'm studying is how immune cells traffic into the central nervous system, either to the spinal cord, which is the sort of bottom heart-shaped thing down here, and then or the brainstem, which is I've taken sort of a snippet of. So I have a mouse model where some sets of mice will get what we call conventional EAE, and they have a primarily spinal cord-mediated disease. So they have issues with like tail strength, like Julie was talking about. They develop limp tail, and then sometimes their hind limbs get weak or paralyzed. And so in the one where you see a lot of infiltration in the spinal cord on the left, those blue cells, the blue dots are cells that are infiltrating the spinal cord. The green is myelin. It's the sheath that surrounds axons in the central nervous system. And what happens is those immune cells get in and they start eating away at the myelin. And that happens a lot in the spinal cord in these mice with conventional EAE. But it doesn't happen as much in the brainstem. There are some cells in there, but for the most part, the green myelin is left largely undamaged. There's another form of EAE we call atypical EAE because it's the one that doesn't happen very often. And what happens in atypical EAE is that those cells actually primarily go to the brain as opposed to the spinal cord. And we're trying to figure out why that occurs, and it only happens in certain subsets of mice, and we're trying to figure out why they decide to go to the brain in that case and not the spinal cord, which you can see here there aren't very many cells and it's nice and green. So my research is focused on figuring out how the immune cells get into one area or another, with the hopes that that might actually translate into certain MS patients that might have certain sets of immune cells that are more likely to go to the central nervous system, and they prefer to traffic to either the brain or the spinal cord, so we can kind of zero in on what cells might be causing the problem. Thank you.
So I mentioned that Angela has a condition called neuromyelitis optica, which is closely related to MS, but it targets the spinal cord and the optic nerves, and it spares the brain. And then there are people with MS that only have lesions in the brain and not the spinal cord. So Josh's work will help us understand why some patients have one part of the nervous system affected more than others. And that's important because we're finding that depending on where the inflammation and damage occurs, one therapy may work better than another. So if we understand this in greater detail, we can begin to customize therapies and get it right earlier than we can now in terms of figuring out what's the best approach in an individual patient. Okay, and now this was supposed to be a video that isn't working, but this is 3D, it spins around, and it's really almost like an amusement park ride. But Kevin, could you tell everyone about this?
Yeah, sorry about the video, the format, something happened. But I did my PhD in stem cells and regeneration, and when I came here, I was still interested in helping neurons survive, or nerve cells survive and recover from damage. You guys already mentioned the optic nerve. This is actually the back of the eye here. This is the retina, which is actually part of the central nervous system, and this is the nerve that would transmit the signals from the back of the eye towards the brain. And I'm working with the eyes in mice to to potentially learn a little more about how these nerve fibers can be spared or rescued and help to grow back. In my work, we have these mice where their nerve fibers are lost. So the bright green here is healthy nerve fibers, and they've been essentially separated or disconnected from the brain. This mouse would be blind. And we are looking actually at how good inflammation, so you guys hear about how inflammation is always bad, but we're learning that there's some aspects of inflammation that might have some benefits. Different subsets of immune cells in the inflammation are actually coming in and potentially promoting the survival of these nerve cells and telling them to start growing again. And we're looking at this, and we're injecting some of these cells specifically into the eye, and you can see some of these right here. This is kind of a blow up of the back of the eye. This is the retinal ganglion cell layer. These are the most, you could say, the most important neuron in the back of the eye. That's the cell that connects to the brain. And this in red and in green, these are these immune cells that I injected. And following this injection, these cells will make contact with these nerve cells, and we see this robust regrowth in their fibers that they start to project again down the optic nerve and towards the brain again. And so this would be more of but the idea is to find some therapies for secondary progressive patients, right? To rescue nerve cells in all patients, but hopefully at some point develop therapies to bring them back and make connections again in the central nervous system.
Thank you, Kevin. So you know, in multiple sclerosis, there's damage to the myelin, which is that wrapping around the nerve fibers, but we're finding that the nerve fibers themselves are often damaged so that they're split apart. And that causes some of the persistent disability in multiple sclerosis. It occurs in relapsing remitting MS, but also in progressive MS. And many of you know in relapsing remitting MS, you do recover somewhat following episodes. Many individuals do. So the body does make an attempt to repair, and it's only partially successful, and ultimately that burns out. But it turns out that even though many of the inflammatory cells initially cause the damage, there are some inflammatory cells that may initiate the repair after the damage is done. So Kevin's work is trying to dissect out which immune cells cause the damage and which may actually do the reverse and protect and stimulate regrowth. And I think he's developed a system which is really going to give us important insights, leading to new ways of stimulating repair ultimately in patients with both relapsing, remitting, and progressive MS. Okay. And the last person who's going to share research data with you is Amanda.
Hello. I work primarily, well, not primarily, I have a mouse project too, but what I'm going to show you is the human data that I've generated being here. My studies are looking to find factors that are in the blood, also known as biomarkers, as Ben had pointed out on the slide, that could help to predict the stage of disease or relapse or even lesion formation. And so we've taken blood samples from a set of patients that are either secondary progressive or relapsing remitting over a year. And in addition to the blood, we have MRI scans and EDSS scores. So these are just a few graphs looking at factors that are produced by blood cells from the patients. The top one is interferon beta, or interferon gamma, sorry. And this is just to show you that there is a higher production of interferon from all MS patients compared to the healthy controls, which is your red line. And at the bottom is IL-17, which is another pathogenic factor produced by T cells in MS patients. And here you can see that in the secondary progressive groups, which is the blue and the purple line combined, that IL-17 is actually higher than in the relapsing remitting group, suggesting that as the patients progress, there may be higher IL-17 being produced by these cells. So this is something that we might be able to just draw blood and check for, as opposed to MRIs and more invasive tests. In addition, we are seeing differences in other factors in the blood, and this is just one example. We see many different ones changing either up or down, and we're hoping to try to put together some sort of algorithm so that we can just take a small amount of blood and run these tests on patients. And here we see that in the factor GROW alpha, that there is a clear difference between the relapsing remitting patients and a rise as they transition into the secondary progressive. So this could potentially be used as a predictor of progressing disease. And then finally, as I mentioned, we take MRI images in addition to the blood, and we look for factors that may be going up or increasing before the onset of a lesion, so that we may be able to monitor patients easier than actually just doing constant MRIs every 6 months. And so we see, for example, in the factor, which one is it? Oh, ENA78, which is a factor that actually deals with neutrophils. So it goes back to what Julie was talking about with the neutrophils, that it significantly increases pre-lesion and stays up throughout the lesion in the patients. So hopefully this work, like I said, will be able to screen all of these from a smaller amount of blood and a less invasive test than what you guys currently do. So.
Yeah, so thank you, Amanda. So this is very exciting because we don't have a blood test that'll tell us if the disease is active or not. We have to do MRI scans, and I think this is the first time I've seen data where a simple blood test goes up at the time a new lesion is forming. And it even begins going up, as Amanda mentioned, the month before the lesion. So this blood was taken the month before, during, and after the lesion, as well as at a time point when there's no lesions forming for quite a period. And so this is the type of work that we're doing, both with humans and with mice. And I just want to thank our young investigators for their dedication and the breakthroughs that they're making. Thank you, guys.